I begin by thanking the organizers for having this meeting regularly every year. It's nice to come and talk here. I'm visiting ICTS for the first time. I'll be talking about the spatial organization of the DNA polymer at large length scales. When I say large length scales, I actually mean micron length scales, and much above the DNA helix. The DNA helix has a pitch of 3.4 nanometers, and the chain width is 2 nanometers. Okay, but I am talking about 1000 times bigger organization of the DNA polymer at scale 1000 times bigger, the micron. So one knows, I'll just remind you that the DNA is made up of 80 CG, and this combination of 80 or CG is called the base pair. The human DNA has around 6 billion base pairs packed in 46 chains. If one takes all these DNA chains, stretches them and puts them end to end, you get a length of around 2 meters, and this is packed in a very small nucleus volume of micron cube. So the question is, of course, how can you pack such highly charged DNAs in a small volume? Some part of this has been understood, and one knows that the DNA is packed around certain proteins called histones. So there are around 20 proteins packed around histones, and these histones, DNAs wrapped around histones form a pearl necklace kind of structure. Now this structure of pearl necklace, they themselves pack and form a so-called 30 nanometer fiber chromatin bundle. Okay, so organization from the DNA helix, which is at the 2 nanometer scale to 30 nanometer scale, is understood to some level, but how in turn now the 30 nanometer fiber organizes itself at larger length scales is a matter of intense investigation. Okay. And at this point I would also try to remind you that DNA is a semi-flexible polymer and can be considered as a semi-flexible polymer, and the Kuhn segment is around 300 base pairs or around 100 nanometers. So the basic message from this is, it is expected that DNA is packed in the nucleus by organizing itself into a hierarchy of mesoscale structures. Now even at the larger length scale, there is experimental evidence that the DNA polymer is not packed randomly. So these are the so-called chromosomal contact maps, and basically what biochemists have done is plot using biochemistry methods, figure out what is the probability or what is the frequency or finding certain segments of the DNA polymer, certain parts of the DNA polymer close to other parts. So what you see in the x-axis and the y-axis is basically segment index. So you take the entire polymer, break it up into segments, and by some biochemical methods, so this is segment index, this is also segment index, this is 1 megabase pairs, and so you are looking at scale smaller than the 1 megabase pair, and these red spots means that certain segment has a much higher probability of being found closer to certain other segments. So the one clearly sees that it's not random, there's a certain structure to be found, certain segments are preferentially found with much higher probability compared to other segments. This is again data from some mammalian chromosome 14, I don't remember whether it's human or not. So there is evidence that it's not random even at a larger length scale. The question we are asking is, what is the origin of such organization? If at all there is organization at a much larger length scale, and what brings about it? And my bio collaborators came to me 1.5 years back and said, if they give me such kind of data for a particular DNA, can I predict the large scale structure for a particular chromosome? So what we hypothesized with Farhat Habib and GP Manjunath is, let's assume that there are a small number of crosslinks in the DNA polymer. Okay, so crosslinks at very specific points along the chain leads to organization of the DNA polymer. This is our hypothesis. How do we choose the very specific points? We have the contact maps, we have the biological information where certain segments are found preferentially to other segments. We take those segments which are found with the highest probabilities or the highest frequencies, what I would like to use the word, and cross-link them. We permanently cross-link them. We want to use only a very small number of cross-links. So if you have a chain and you put a large number of constraints, of course it's going to be structured. But we put just 1% of cross-links, 1 to 1.5% of cross-links at very specific positions and ask the question whether it gets organized. And we also compare, if we put an equal number of cross-links at random positions along the chain as different from specific positions, what kind of organization do we get? Does it get organized at all? And so on and so forth. So this is the basic question, and my job was to figure out with these constraints, can I predict the organization? How do I model? So I look at DNA as a polymer. In particular, I took the E. coli DNA and another bacterial DNA, Colobacter crescentus. They are known to be ring polymers. I can use the bead spring model of the polymer chain. I can consider it to be a flexible polymer chain because basically there are 4 million base pairs which I model as 4642 monomers. So one coarse-grained monomer in my bead spring model corresponds to 1000 base pairs, which is well above the persistence length. I use small excluded volume interaction, and at very specific points along the polymer chain, I cross-link by using a harmonic spring potential. For comparison, I also cross-link monomers chosen from a random distribution, an equal number of cross-links that I use in the biological modeling. I already told how I choose my cross-links. For the Colobacter crescentus, by choosing different thresholds, I can have either 60 cross-links or 26 cross-links. For the E. coli, by using a similar threshold, I can have either 82 cross-links or 27 cross-links. And the number of monomers for CC and E. coli are 4,000 and 4,600. So I'm really talking about 1% to 1.5%. So how do I decide whether they get organized? So I start with very different initial conditions, given a set of crosslinks. So given suppose 60 crosslinks or 82 crosslinks in these many polymers, I start from very different initial conditions. I allow the system to relax. So when I put different initial condition, I just make them into a circle or a square or a cube without keeping any constraints about how far these crosslink monomers will be, and now allow them to relax, 9 independent runs, and after it has relaxed, I measure structural quantities and compare across 9 different runs. Starting from very different initial conditions, if they show same structural properties, I would say that, okay, the crosslinks are leading to some sort of organization. But now the big question is, how do I determine the structure of a polymeric chain, which is supposed to be anyway like a random walk, of course with some crosslinks. For that, I define some quantities, or I figured out that, one thing one can do is basically calculate the radius of gyration and ask whether a particular crosslink is to be found towards the central part of the polymer chain, the middle part of the polymer chain, or the peripheral part. So if this is the ring polymer, I can also divide it up into 80 segments, arbitrarily chosen, and ask whether the center of mass of the segment number 1, segment number 2, or segment number 3, whether they are found in the interior part, the middle part, or the peripheral part. Then we can ask questions like whether a particular segment, now I'm talking about 80 monomers, so this is much larger than the length scale at which I cross-link the monomers. So I can ask whether a particular segment 1 is found close to segment number 2, 3, 4, and so on and so forth. Furthermore, so this is basically like the G of R, but at a much microscopic scale. So I am asking whether certain segments are found far away or close to each other. I can also ask whether a certain segment is angularly placed in the same hemisphere. So these are the quantities which I use. Here I have calculated the radius of gyration. Yes. Here I have calculated the radius of gyration for the E. coli. For the larger number of crosslinks, sorry, this is the smaller number of crosslinks. You have a higher radius of gyration, and here you have a, when you have higher number of crosslinks, it decreases. This is for the random case, the decrease is significantly more. Okay, so from here I would say that even with a smaller number of crosslinks, maybe they are already some bit organized. For the biological case, when you put in more crosslinks, the decrease in size is not so much. Here I have basically so spatial location of a particular crosslink, whether they are in the inner region, the middle region or the outer region. These are data for 9 different runs, and I check whether from different runs I get the same organization, whether the same crosslink, and I can do it for the segment also, whether they are found in the inner, the middle and the outer region. I have also calculated Pearson correlation functions, and I see that data from different runs match, which means that across different runs a certain segment or a certain crosslink is found in similar regions. This is data for random crosslinks. So here what I have done is, this is biological, using the biological crosslinks from different runs. This is the segment index. This is also the both on the x-axis and y-axis, one has the segment index. And what these color spots show is the probability of finding a certain particular segment close to each other. If they are bright, it is basically a higher probability of finding from close to each other. These are from different runs. You can see, compare these 3, there are 9 of them, that they are very similar in shape and structure. They get similarly organized. The same segments are close to each other from different runs. This is data from random crosslinks. I have equal number of crosslinks but randomly chosen. They look similar to each other, but you would see that the pattern of the bright spots are much more diffuse compared to the one with biological crosslink. This is data for some other, for the other CC bacteria, and you can conclude very similar things. This is comparison of biological data, random data. Finally, putting this and the angular correlations together, I can predict the 2D structure for the E. coli and the Colobacter crescentus. So I've divided into 80 segments. I can tell which lie in the middle, which lie on the periphery, which are opposite to each other. This is our theoretical prediction. Can be cross-checked by experiments in the future, which I hope my bio collaborators will do. And with that, I conclude. My basic conclusion is, it seems that crosslinks at very specific points along the contour of the polymer chain determines global chromosomal organization. And this work was carried out with Tejal Agarwal, my PhD student, bio collaborators GP Manjunath, Farhat Habib, and I thank you for your attention.
So your chains are self-avoiding, right?
Yeah, self-avoiding.
Does that make a difference?
We are doing a systematic study by changing the excluded volume interaction.
Because given your beads are like 1,000 or some 10 to the power 6 base pairs, yeah. I would imagine them to be self-interacting. Right? I mean,
so at the moment they have excluded volume interaction, but a systematic study of, as I increase the excluded volume, how do things change? And also if I put in slight attraction to model collapse, this will be the next stage. First stage, only excluded volume interaction plus crosslinks. So this is relevant, and this is under way. There can be DNA binding proteins. Actually, there exist DNA binding proteins, and they do hold it together. Whether they do it to organize it for large length scale, we don't know. But putting crosslinks is not an absurd hypothesis. No, I don't suddenly switch it on. Initially also, so I put them very far apart. I have a very weak kappa for the crosslinks. I slowly increase it and allow the system to relax. So the crosslinks are there. So it will relax to its final configuration due to the presence of crosslinks. Else every polymer would take a different shape. I mean, that's what we expect. That's what we get also with the number of crosslinks.
Initially, you mentioned different length scales. So these crosslinks are at what length scales?
They are at the rate 1000 base pairs. The experimental data, the resolution that we get in the contact maps is for 1000 base pairs, kilo base pair, and our monomers are also at kilo base pairs. One considers this DNA without histones and so on and so forth, that would be order of 100 nanometers. We are starting from 100 nanometers.
Okay.
The modeling is at the level to 50 to 100 nanometers.
So hypothetically, if we have crosslinks at a lower or higher length scales, so would the final structure be still of the micron length scale?
At a smaller length scale, well, you're basically holding two parts of it together. I mean, if you go into further details, this shouldn't matter. But we don't have data. We have biological, so there's biological input that is at the kilobase pair level. That is what we have put in. We'll try out for many other polymers, many other DNAs which we shall model. Those I have to get the biological data, then only I can confirm. This is 3 dimensions. No. It was in 3D. It's a Monte Carlo 3D.